right? In general, or monoallelic. The monoallelic expression could be of two types, but largely three types basically. One is that so one could be random stochastic, which means that any of the allele could be expressed stochastically, right? Not both of them. And sometimes, in some cases, both of them get expressed, right? So random one could also be sometimes viable. So you have another one which is mitotically stable, which means that at some point during proliferation, a decision is made that which allele will be expressed. It could either be maternal or paternal. But once that random decision, so this is of course random. So initially it's random, right? So, initially or established randomly. There is no choice of maternal or paternal. Either could be selected. But once selected, it is mitotically stable, which means that if you are deriving a clone of cells, in that particular clone you will have only one allele expressed and that allele is fixed, either maternal or paternal. But the choice, whether it's maternal or paternal, could be random. It's a kind of random monoallelic, but it's not completely random. So the choice of maternal and paternal is random, but the inheritance, epigenetic inheritance, is not random, it's stable. Third one is parentally fixed. Okay, so what is the example of this? Do you know any example of mitotically stable?
Chromosome expression.
X inactivation is one example. Okay. Now X inactivation is constrained only on X chromosome. We also have something called autosomal monoallelic expression. So it's not only constrained to X inactivation, there are other loci also. Up to 20% of the genome could be monoallelically expressed from autosomes. And then we learn parentally fixed monoallelic expression where the choice whether it will be maternal allele or paternal allele which is expressed is parentally fixed. This is your gene imprinted. Imprinted gene expression, genetically imprinted. Don't confuse that's why I use the word genomic imprinting because there is imprinting in ecology also. The moment I say imprinted, it means we are talking about the expression of DNA methylation. So, imprinted allele is always silence. If I say maternal imprinting, it means maternal allele is silence. If I say maternal expression, it means maternal allele is expression. Imprinting is synonymous to silencing. So we talk about the example of IGF2. So let us zoom in to IGF2 locus. So it is located on chromosome 11 in human and chromosome 7 in mouse. And here is the structure. We have IGF2 gene. Another gene which is long non-coding RNA basically. The ancient most this is actually conserved in all mammals. This H19 RNA is conserved in all mammals. And in fact, this is the one non-coding RNA which has been known for ages. One of the first most non-coding RNA, except after XIST on X chromosome, is the H19, which is known as long non-coding RNA. And this is also imprinted. So H19 is expressed from mother, IGF2 is expressed from father, from paternal. And there is an in-between region over here, very important region, which is called imprinting control region. Just like you have LCR, locus control region, or beta globin locus. So you remember you have several globin genes in the cluster. Okay? And some globin genes are expressed in the embryonic tissues, some are expressed as standard development during postnatal development and in adult individuals. So these 3-4 globin genes are actually controlled by one control region, one enhancer region. That these days people call it as super enhancer. So in the earlier days we were some fancy rebranding of previously known phenomena. So we have number of enhancers together, binding sites of factors, call them as LCR. So it access, it loops over certain genes in embryonic tissue or their expression stage, and then it doesn't interact with them. The adult tissue interacts with other globin genes and different globin genes which are expressed in embryonic stage and in adult stage. So likewise, you have ICR, which is imprinting control region. If some new scientists will get to know there's something called ICR, they will find a name. So it has several 4-5. Of course, there are other sites, but there are at least 4-5 that characterize CTCF binding sites. This is, of course, a non-coding region, and then you have an enhancer upstream to H19. Now, let us see what happens on the maternal chromosome. Let's say this is maternal chromosome. And we know that IGF2 is silenced and H19 is expressed. So how is that happening? What happens is that you have your CTCF coming over here. The reason I made it in a elongated form, I want to show this as a barrier, as an insulator. So now what happens is that this enhancer has access H19. So H19 is expressed. And this is not having access to IGF2 promoter, so this gene is silent. This is what is happening in maternal. Likewise, there are examples of other imprinted loci also. This is one of the first most site where the insulator function of CTCF was characterized. It is insulating this enhancer to access the IGF2. What happens in the paternal chromosome is that you have some kind of, not some kind, this is actually methylated. This ICR is methylated, which means that, and not only this is methylated, the upstream region of H19 gene is also methylated. So the promoter, which is over here, is also methylated. So this whole locus is methylated, then CTCF cannot bind, right? So CTCF. Not able to find detachment. So now what happens is, since H19 promoter is methylated, H19 is silent now, but this enhancer can now access the lack of insulator binding. It is able to access the IGF2 gene and it is expressed. And this one is silent. This is happening. You might wonder how this is happening. First of all, you need to understand something which is not well understood how this maternal paternal is recognized inside the nucleus or the transcription that which allele to the nucleated, which allele is PCH defined. Some people have some idea that it's actually demarcated by early and late replication. Generally, the highly active genes or active alleles are early replicating and silent alleles are late replicating. Okay, however, in case of imprinted genes, it's other way around. So the silent allele is early replicating and active allele is late. Okay, sorry. It is always the maternal allele which replicates early and paternal allele replicates late. No matter of what is the status of that gene. In this case, for example, IGF2 is silent, but this will be replicating early. Okay. So regardless of the transcription status, it is always the maternal allele which is replicating early. Just give me a minute, I think I got it wrong. Let me check it. Whether it was maternal or paternal, I'm just missing. Sorry about this. Looking into some notes that I have support now. The maternal allele should replicate late. That paper and actually so it's the maternal allele which replicates late and it's paternal allele which replicates early. Now, interestingly, this ICR, if you have mutation in the CTCF binding site, of course you will have biallelic expression. It turns out that if you have mutation, the maternal allele starts replicating early. Not only the IGF2 and H19 gene, all the maternal copies of imprinted genes in rest of the chromosomes also. Okay, this is mind-boggling observation. It needs more work to confirm and establish the significance of such thing. So this is one way of marking maternal and paternal imprinted genes. Okay. Maternal or paternal copies of imprinted genes. So which means that factors which are regulating imprinting, they recognize replication timing as a hallmark for recognition. Okay, again, all these are at hypothetical level, but there are evidence that they replicate asynchronously, and which is fixed. It is also fixed currently. Okay, now you might wonder how does this happen? The genome is not linear like this. It happens in three-dimensional way. That 3C, when it was innovated, some of the very first experiments were on this purpose. So what happens on the technical performance is that you have structure like that, where you have something here. Why? Because we have answer in the same complex. There will be another loop here, so I don't want to complicate it. However, you have ICR here, okay, and there is a upstream DMR region, differentially motivated function, just like ICR. ICR is also a DMR. DMRs are ones which are regions which are differentially motivated QRDs, like CTCR region is. So, what is a big complex? How can I talk about H90? While this region would have inactive you're making some kind of 3D structure which is two separate complexes of active and inactive alleles. And what happens when you have this interaction and you have a structure like that? Your H90 is here. Silent. Why? Because upstream region is motivated. Right here is here. Now you have your enhancer over here, and you have IGF2 gene here. This is being transcribed now. Why? Because the enhancer is accessing this. There is no CTCF binding. So it happens through looping, again specific looping. Now, this has important consequences, and you will see that a little later when I talk about the developmental deprogramming, epigenetic deprogramming. Okay, so this was all about imprinting, and we already discussed some of the theories of imprinting. Parental conflict theory, the most accepted one. Sorry, the second one was showing the wrong finger. The second one was maternal offspring adaptation. Third one was parthenogenesis, barrier to parthenogenesis. Ovarian time bomb, which is very similar to parthenogenesis, just that leads to seeds. You don't have any dead structure. And then you have cytoplasmic conflict between mitochondria and internal hormones. Selected. Minimization of variance of genes, because these genes are dosage sensitive. So, and then there's another theory on genome defense thing, and that is based on the observation that all the foreign DNA that we have in our genome, like chromosomes, they get silenced through methylation. And it turns out that most of the imprinted genes are actually enriched near transcriptomes. And the main mechanism of regulation of imprinting is methylation. We also have transcriptomes out here. So it turns out that it's the byproduct of genome defense against foreign DNA. So, however, each of these theories have their own subcomings. For example, to support each of these theories, you need to show that gene functions are actually associated with your theory. For example, if I say imprinting is able to resolve the cytonuclear conflict of mitochondria and nuclear DNA, then we need to show that the eternally expressed genes in the nuclear DNA are actually associated with the coordination with the mitochondria. Right? Quite often, people don't have significant numbers with the relevant genes, which is why parental conflict theory and the maternal offspring adaptation is well established. The maternal offspring adaptation is established because of the very striking observation that most of the genes which are imprinted in placenta are actually maternally expressed. So IGF2 gene and many other growth factors and developmentally regulated genes are involved in growth, right, and the development of the embryonic development and post-development, which supports the theory of parental conflict. So again, these are all theories. Okay, so let's move on now. So let's talk about something called epigenetic reprogramming.
निकालने के लिए और 2 घंटे बैठना होगा ना
1.5 घंटा
हाँ तो
मैं निकाल दूंगी
कितने टाइम तक रहेगी
ऑटोक्लेव निकालना है
हाँ
ऑटोक्लेव
बॉटल से खा लेगी
वही तो निकालना होता
So
है
ideally,
नहीं,
you
बात वो
should have
निकालने
taught
के लिए कि वो बॉटल
another classes
ऑलमोस्ट 2
before
लीटर की बॉटल्स है
the genome and all that. But if it has been taught, we can stop here. If it is not taught,
चलो कोई बात
then I can go ahead.
देख लो
Was it taught at
तो ये
some
जैसे
point
हम उसमें ऑटो उसमें पानी डाल रहे हैं
in any
अच्छा
system?
टाइप करता हूँ लिख देता हूँ क्योंकि झंझट ये है कि मैं इसलिए करने नहीं जा रहा अगर कोई गड़बड़ हो जाए फिर कल खाली लास्ट है ना फिर से बोलेगा फिर से जाके लेके आ जाओ साइड इसीलिए नहीं अरे ये फॉर्म में कुछ नहीं है ये फॉर्म मैं बोलता हूँ क्या है मैं देखा अभी इसमें मेरा नाम लिखना
I'm
होगा
going
इसमें
ahead
लिखना
anyway.
होगा
There is no response from the students.
तो मतलब ये एक ऑफिशियल डॉक्यूमेंट है जिसमें मेरा बिल का इनवॉइस नंबर जाएगा इसके
So,
साथ मैं बिल
what
अटैच कर
is
दूंगा
epigenetic reprogramming?
बिल के लिए और
Epigenetic
बाकी
reprogramming during the
अच्छा नहीं पड़ेगा ना
हाँ
now development is of two types.
अच्छा
That we talk
एप्लीकेशन
about
नीचे
mammals,
साइन कर सकते हैं ये साइन कर रहे हैं
very little contact. So you have germline development and of course embryonic development.
नहीं ये इसमें ना बिल में
Embryonic and
बिल में क्या है कि
post-embryonic or
इसमें
postnatal.
नीचे सिग्नेचर ऑफ द स्टूडेंट
So let us say I'm going to plot final
ये
x
इसमें हो
axis
जाएगा
and y axis is
तो मैं
some percentage methylation, percentage methylated DNA.
तो अच्छा एक बार बोल लो उसमें क्या लिखा था मतलब एप्लीकेशन में क्या लिखा था इन्फॉर्मेशन क्या क्या था उसमें
The genome. And we are going to plot two trajectories, one for maternal and other for paternal.
क्या दिया था वो सेमेस्टर फिर दिया था क्या
Let's start from the fertilization.
हाँ हाँ वो तो है
So the point is, let's say, this side.
तो
Suppose
हाँ
where the size starting is small. Okay, those who tells you in some courses in ICMOI or otherwise that the
ओके ओके
in the nature cell size
तो
is
ये भी ठीक है
constant
इसमें लिखा है कि दिस
and cell number is varying.
पेमेंट हैज बीन
That
पेड बाय
whether
द
you
स्टूडेंट
talk
फॉर
about
ऑल द
mouse or elephant, it has more cells, but why didn't nature choose for bigger cells?
इसके नीचे ये साइन कर रहे हैं
Okay, so they
सब ठीक है
lead to a claim, the broad claim that the cell size is constant. Cell size is continuous.
ठीक है ठीक है
The same organism
ओके
you
ओके
have
मैं इससे ही देख
multiple
लेता हूँ
cell types, and that's because you have
आएगा
something
तो
called adaptive selection. Each cell type has its own function. One of the examples, look at the size of oocyte and sperm. Huge difference. You can look at your spinal cord. Right? So there is one big axon going out there. Likewise, you have various kinds of cells in plants which have different function, like stomata. Those cells are quite different. So let's look at the trajectory. So let's say there's some methylation level here. And I have shown sperm on the top because the methylation or the compaction of sperm DNA is very high. And there is a reason why we have very compact DNA, because the proliferation of sperm is very rapid. So it's more prone to DNA damage. Looking for multiple reasons, not only the proliferation, the meiosis, the activity is also high. It has a flagella and it has to move. Also, a high number of mitochondria, oxidative damage can happen. So, chances of various damages in DNA is very high. So, nature has found a way to really condense the DNA through methylation and proteins, not histones.
मतलब अभी सारा पढ़ा है मैंने कि मुझे
So,
क्या क्या लिखना
DNA
है वगैरह
percentage DNA methylation of sperm is quite high as compared to
अच्छा है वहाँ पे। तो मैंने
X
ऐसे पेपर
oocyte.
वगैरह थोड़े पढ़े हैं बट एक भी शब्द नहीं लिखा है
So
मैंने
you-
अभी तक मैंने सारा
So what happens is
Saturday,
the
Sunday
first
के
thing
लिए
which happens is that after fertilization,
हाँ
the methylation level will start dropping. So it turns out that the methylation, so let's say fusion has happened. Now we have this station where होता है. So what happens is the following: the DNA of sperm genome start getting demethylated very rapidly, and this is active demethylation. So you would need glycosylase and TET3 enzymes. Are you aware of these enzymes? And the demethylation of the paternal genome, so fertilization has happened here at this stage. Okay, so what I'm reporting is within same cell. So fertilization is somewhere here, or actually it is already fertilized. Okay, so actually it's already fertilized somewhere here. This demethylation happens after fertilization. So let's say fertilization has happened here. Okay. And you have the maternal genome is passively
हाँ
demethylated,
अरे वो
and
कंटिन्यूसली
maternal
सबमिट
genome is actively demethylated for the sheer fact that
इसमें क्या जो
you have
पीडीएफ है
more
मेल में
DNA to be
प्रिंट करके
demethylated
देना होगा ना उसको
in paternal genome. You need an active demethylation. Active means you need
जो है
glycosylase and TET3 enzyme. Active demethylation has to happen.
तो
So when I was doing my PhD, then there was a saying that there was various DNFTs were known,
जो पीपीटी
the
है
enzymes which are methylating CPGs. There was no enzyme known which is demethylating DNA, but everybody knew that it has to
अच्छा तो
exist.
इसमें ना इनवॉइस
Most
नंबर ये सब
people have evidence of active demethylation, but it was not known. Nobody has
मेरा
identified any
मेरा
demethylation.
मतलब खर्चा
And
हुआ तो
anybody who
मतलब मेरा जो बिल था
discovered that we
जो
get a
कॉमेंट
fine.
से लिखा है पेमेंट किया था
It turns
So
out to be a
इसमें
cell
आप
paper, and they didn't discover a demethylase enzyme. They discovered glycosylase enzyme,
अच्छा तो मैं उसका जो है वो
indirectly
था ना
demethylates the DNA. So that is happening in the paternal genome. So the maternal genome is passive demethylation, which means that during replication, the existing demethylated DNA is getting diluted over time. There is no new methylation happening. So whatever methylation was there in the hydrocarbon site is now getting diluted with methylation. So it's a slow process. You would be surprised that since the paternal genome is actually
अगली बार ले लेगा
compressed one, and most of the activity is actually controlled by maternal genome, right? Maternal
That's
genome
fine.
is making paternal genome to get demethylated activity. So the enzyme to demethylate paternal genome is actually coming from
25 तक
paternal
लास्ट
genome. So this
हाँ।
genome is actually demethylated. It's the maternal genome. This is called maternal zygotic control. Okay? So initial development, initial processes in the zygote is actually controlled by maternal factors. A number of those factors out there. Most of them are transcription and chemical factors. Okay, so somewhere, during this process, you will have the 2-cell stage, 4-cell stage, and all that, right? And you reach at around mouse, this is around 3 days post-fertilization, 3.5 days post-fertilization, when you reach the blastocyst stage. This is your blastocyst. And you know that most of the genome in the blastocyst is actually the inner cell mass, ICM, right? ICM have demethylated DNA. But this is a trophoblast which remains methylated. Okay, it remains methylated. So this, if you plot about the trophoblast thing, then your trajectory would be different. Let's say this is the average trajectory coming up. now. So trophoblast is against right. So rest of the cells, they are working to ICM. DNA will be demethylated except trophoblast, and then this will again develop into there will be DNA methylation program established, right? Why? Because now you need cell type specification, differentiation. So
As
this will
तुम्हारी
get,
matches?
this will get methylated again.
All the way.
Some of you might wonder why this is coming down, going up. How is this happening? Anybody,
कौन
how
है
is this
मतलब किसके
happening?
साथ है?
How did it go up again? So you have this development going on, right?
Batch?
कुछ PS नंबर MS नंबर लिखा होगा
अच्छा WhatsApp ग्रुप में.
So a lot of the methylation, of course, now
हमारे
not
batch
whole
में
genome.
दो
This
हमारे-
y-axis doesn't go to 1. It's not like 100% of the genome is methylated.
But
Nothing will be
मुझे नहीं एक उसमें से एक पे
transcribing.
definitely
So
है दूसरा.
let's say
वो भी सब
proportion of the
तो
max
होगा,
possible
पता नहीं
methylation.
होगा,
Different cell types will have different expression profile. The genes which are not supposed to express in your brain will be silenced in brain. The genes which are not supposed to express in your liver will be silenced in liver. So it's the methylation as well as other histone modification which will define the trajectory of various cell types. The cell type
Amar?
specification is established through
Anya?
genetic modification. So the total methylation level will go up here. So this is around time point is 7 days or 7.5 days in mouse. Okay, so it remains this methylation level remains same in the somatic tissue, like your brain, your liver, your lung, your skin, your eyes, your teeth, your tongue, everything is these are example of somatic tissues. But there will be some cells which will make your germline cells. What are your germline cells? Sperm and your oocytes, right? So those cells, the cells which have cell fate of certain cells in this particular stage of the embryo will form something called primordial germ cells. So now a lot of the cells here are actually methylated. So these cells, let's say somewhere here, they have the cell fate of making the post-primordial germ cells. What are primordial germ cells? Primordial germ cells are germline stem cells. So just like ICM is your embryonic stem cells, your PGCs are germline stem cells. Germline stem cells will also get demethylated. If you talk about PGCs, the cells which are going to form PGC will get demethylated. You have cells here which are PGC, primordial germ cells. So they will be demethylated. Why? Because now they have to differentiate differently in males and females, right? In males they will make the sperm cells, in females they will make the oocytes. Now again we need to use two colors. So it turns out for males, this is important here. For males, let's say, so this is PGC, and then I need to actually get the time point correctly. Somewhere here.
मुझे लगता भी नहीं है कि वो ऐसे सीरियसली कभी खेल रहा है, ऐसे ही खेल रहा है.
Which means that right now what you see here is the embryonic development. Okay? So you will have postnatal development after this. So you will have postnatal.
सिंगल है तो कितना ही
एक्चुअली होता है 3 सेट मैच होता है
And then during the postnatal development, you will have another stage, important stage, which is your puberty.
15-30
Then you have fertilization again, back to zero, right?
तो 6 का दिन से
हाँ.
So what happens is that all the so in the male germline development, this is your germline development, male germline development. Genome gets methylated by the time of birth, most of it, and it remains into this stage from PGC to birth is your mitotic arrest in male. So all blue color belongs to male. Mitotic arrest, during which it is getting methylated. But slowly it's getting methylated. The DNA in the cells, which is you can call them as pro or pre-spermatogonia. Okay, here is the spermatogonia, spermatozoa, then sperms. So by puberty you will have meiosis started, right? So this part would be your proliferation.
सीनियर हो गया ना वो तो जो एक साल बाद कैसे
Spermatogonia, prospermatogonia, whatever you call it. Proliferate. Proliferate means growth, right? Proliferation.
तुम्हारे साथ अच्छा है हमें तो एक साल बाद दिया
And here will be your
हाँ तो फिर यही
so rest of the life
नंबर डाल दो
the lifespan will have the meiosis, right? So, which makes these sperms, but it starts at puberty, right? When the meiosis or the sperm formation starts at puberty. Okay. But something strange happens for the female germline. So first of all, this whole thing is delayed. Okay.
24 में से 20,000 ट्यूशन
Goes like that. So what you have here is this whole thing is actually your meiotic arrest. From PGC to puberty is your meiosis 1 arrest, and from puberty to fertilization is your meiosis 2. You remember the oocyte actually starts dividing. The final meiosis happens after fertilization. Okay, interestingly. This is the fertilization stage. So meiotic 1 is a very long arrest. Okay, so you have oocyte here.
मेरे पास 18,200 इतना भी कम नहीं है
Oocyte here. But that is arrested in 4N DNA, not diploid 4N. So which means DNA is replicated, but it is arrested in 4N stage for so long, can remain arrested for almost 40 years. Okay. In total, if you count M1 and M2, remain arrested in meiotic arrest for almost 40 years. So every month during the menstrual period, menstrual time, you will have some release from M1, okay, for few oocytes. Okay. Why nature tries to preserve these oocytes? Okay. So it is not releasing all the eggs together at one go. So very few eggs, and nature is releasing them every month. So from puberty to menopause, you will have this monthly release of from the meiosis 1. Okay. And after fertilization, you will have release from meiosis 2. So that's the way it happens. Now you might ask, why is that so? In the female germline, and why do we have this time delay difference in male and female? First of all, we shouldn't be comparing them head to head because they are in different environment right now. So the male germline environment is different as in males, right? The female germline environment is completely different. It has different environment, not only the physical environment, the hormones are also different. It needs utilizing hormones for release from M2. From M1, it needs, I think, follicle and other several hormones, gonadotropin hormones, which are needed to get release from these arrests from meiosis. Likewise for male meiotic arrest. Now, first of all, they are in different germline environments, so you have to get comparison. The second thing is that nature tries to preserve these oocytes until the individual or the organism is physically ready for reproduction. It doesn't want to release beforehand. There is reason that number of inserts are very good. So it keeps it arrested for a long time, try to preserve it until the person reaches the puberty stage when they are ready for physical reproduction. Okay, so there are strange consequences of this thing. Meiotic long meiotic arrest in female germline. One of the consequences that since it is arrested, okay, it is not replicating, so all the methylation which is happening has to be de novo methylation.
ITM में ट्यूशन फीस
You remember how the replication happens in a semi-conservative manner, right? So when the DNA is replicating in proliferating cells, the cells are dividing, it recognizes hemimethylated DNA and DNMT2 will methylate the other strand, the missing side strand. That's one mechanism. It's not de novo methylation. De novo methylation is that the whole DNA, both the strands are unmethylated and it was supposed to methylate it. Since the cells are arrested in 4N stage in female germline, you have no possibility of the semi-conservative thing, semi-conservative mode of DNA methylation, methylating the hemimethylated DNA, right? So the cells needs to methylate in female germline. The DNA has to be methylated by de novo methylation. So experimentally, if anybody is interested in studying the de novo methylation and demarcate it from the other methylation, hemimethylation mechanism, you need to study oocytes. Okay, if you take any somatic cell, that will have combination of both. So you can't demarcate which one is happening to which mechanism. Okay, so number one, and it has 4N DNA to methylate, which also takes time. Okay, and it is happening to the methylation, which is more rarer mechanism of methylation, unlike the semi-conservative or hemimethylated mechanism that is more common, more frequent. That will also need more time to get methylated. May not be these many years, but still more than the other mechanism. Right, so this is what the epigenetic reprogramming is. Okay, so there is an escape here from it. For example, everything will be demethylated in PGCs. Why? Because it has to remove the somatic marks. What are those marks? The genes will be, some genes will be methylated, others are unmethylated. And you don't want those marks because you need to make germline cells. You need to make sperms and the eggs. Number one. Number two, even the imprinted marks, which are allele specific, has to be erased. Okay, because you have a certain thing, let's say you have maternal genes expressed and paternal genes are silent, maternal copies are silent for this particular allele, and you went to PGC now with this methylation status. So now if you make, let's say, sperm, there will be one sperm which is having maternal gene expressed and other is maternal gene silent, so you have differences among sperms in a population. You don't want that, you want to clean everything. Right, and re-establish your imprinting marks. Okay, so everything is cleaned up in PGCs, and imprinting is established during this time, during just at the onset of meiosis. Okay, just at the onset of meiosis. Here, not overlap. If somebody asks you when the imprinting is established, it is at the onset of meiosis. So the male will be here, right? And in females, actually the meiosis already started over here, right? Somewhere here. Meiosis 1, it is arrested, doesn't matter. So imprinting will be started at the onset of meiosis. Okay. Number one. Number two, once it is established in your germline, okay, it cannot change further. So which means that after this is established here, it will remain static after fertilization. And so if I now plot trajectory of DNA methylation of imprinted regions, it will be like this. It will not get demethylated during development. Why? This trajectory is, so let me put it in different way. So this is schematic dotted line. So imprinted DMRs, imprinted regions. It remains methylated because they have role in development. If they get demethylated, then both the alleles are expressed. Then what is imprinting? There's no imprinting. Printing has a role during development, and so it has to maintain those imprints. So except imprinted DMRs, everything gets, most of the things get demethylated during embryonic development. There's another exception here, and what is that exception? What are the regions in the genome which will not get demethylated? If they get demethylated, there is a danger. Anybody?
So transposons?
Yeah. So all the repeat element transposons are exceptions. Okay. So we could say that particularly during germline development, specifically during germline development, embryonic development is of course the level of demethylation would be lower for transposon. So in both cases, transposons, particularly the notorious ones, there are a lot of transposons which are actually adapted by our genome as a regulatory element, which will have demethylation here. But the ones which are notorious, the moment they get open, they will start popping themselves, retrotransposons. They have to be kept silent. Let us say for some reason they are not kept silent and by some errors they open up. Is there any mechanism to silence them? Is that mechanism exists in germline?
IRNS?
So RNA is expressed in germline specifically. So they are meant to repress the erroneously express retrotransposons. So transposons are exceptions here. Apart from it, you will have most of the following this trajectory. Now, I only plotted percentage methylated DNA, but you can extrapolate this to other histone. For example, I want to plot how do I take note of this? Anyway, so let us take one graph of here. Let's say I want to plot, I didn't use green colors. So let us say I want to plot the distribution or the trajectory for active histone modification, let's say S3K4 methylation. So it will be inverse of this, right? So it's going to go like that. Right? So if I plot S3K27, it will follow the DNA methylation. S3K9 methylation at glucometry necessary, it will follow the methylation. Right. So you can extrapolate other histone modification. I mentioned in the other class that briefly talk about what happens to higher order chromatin structure. So this was your or gate modification. They will roughly qualitatively follow this pattern, but quantitatively, of course, they will be different. And qualitatively, there are different implications also in overall pattern. So, if we talk about the ICK, after fertilization, the genome for both maternal and paternal will not have tags. So tags will start appearing after some time, and they are really resolved during 3 to 4 days post embryonic development, post fertilization.
कितने और बेबी है
5.5 है ना
They become very stronger.
क्या करना है इससे 10
So that's what is known during development. Cats are actually formed, they are following this demethylation pattern. There is some minor difference in maternal and paternal. Maternal shows certain cats, paternal genome shows more cats in the beginning and likewise. There's a minor difference, not a great deal of difference, but there is a difference. So this was all about genetic reprogramming in one slide. One has to really elaborate this in a week's time. Because I didn't go through about germline development in great detail, what is primary oocyte, secondary oocytes, and so on, to get the better idea of this genetic reprogramming. I wanted to briefly talk about, we're running out of time. Okay, by the way, one important thing, because diseases, we didn't talk about diseases. Most of the complex disorders, development-related disorders, are actually result of errors during these phases, right? Let me highlight these phases. During these phases, when things are demotivated, and during the process of this cell division, meiosis process, right? So if you have any error in these phases, you are predisposed to developmental disorders. Okay. Now think about this and think about transgenerational inheritance. If things are being erased twice, epigenetic modifications are erased twice, once during germline development and once during embryonic development. Then how come we keep the epigenetic modification from one generation to another generation? The mitotic stability and meiotic stability, let's not talk about that. Look at this figure now. You have two phases when the epigenetic marks are erased. Then how are they going to next generation? When we talk about transgenerational inheritance, if you know the overcarriage study, right? So it turns out that if my grandfather has seen famine, he was deprived of food, he was having shortage of food, deprived of food, and I am more prone to die. So maybe it's likewise. And when was that famine that my grandfather has seen or my father has seen? If my grandfather has seen famine during his puberty time, when he was 18-year old, okay, so you know that this is the time when imprinting is being established, right? This is the time when imprinting is being established in male germline. Right? This is the time, puberty time, when onset of meiosis, when imprinting is being established. And you get error there, okay, which means that something has happened to imprints, which is being transferred, which is being further transferred to the next generation. Those errors are happening here, and those phenotypes, those erroneous epigenomes will be inherited from this trajectory, right? To me, isn't it? But then they are also again erased during PGCs, right? They are being erased and erased. So what's happening? What do we mean by transgenerational inheritance? Okay, now let us. So this was male thing. When my grandfather was in his teenage, he was deprived of food, there was famine, I got predisposed to diseases. And there is evidence, it has happened, there is data out there. For females, it's the other way around. So meiosis starts even earlier. So when, let us say, my grandmother, okay, when she was in womb, okay, she was not born yet, she was in womb and there was some error. She didn't get good food. She got a lot of food, junk food. And then granddaughter or the grandson will have some predisposition to diseases. Why? Because the meiosis has started early in females, right? When they were still in the womb, before birth. Okay. So there is a paper, and I think I have given this paper to some student, over-calyx study. We see that paper during that. Now my question to you people is that if this erasure is happening, even the imprints are erased during PGC development, right? And how this conservation inheritance is happening? The correct answer to this is that we do not know entirely what is the mechanism of conservation inheritance, but I think it is happening through the regions which are escaping this trajectory. It has to escape. Transposons are one region which are escaping this trajectory. And there are other hypotheses also. Okay. There can be certain regions which are not properly paired. Okay. During homologous pairing of there are certain regions by error they are not properly paired, they get methylated. And this methylation is not erased during this development. So we precisely do not know, but one thing seems to be very logical and clear that it has to be regions which are escaping this trajectory, which is escaping the demethylation. Okay, so I will end with the X inactivation, and I hope most of you know what is CalicoCat. Anybody to look at the cat will have those patches, right? Patches are orange and white and like that, right? And this is, you know, that this is female cat, and I can tell you that my grandmother was pretty good in that, and in fact people in those ages knew intuitively, they didn't know science about it, but she used to tell me we had a cat at our house and we got kittens, and one of the kitten was spotted, and she said she's female. My grandmother told me when I was kid she is female, and I was wondering how does she know it is female? It's very difficult to make out from kittens which is male and female. And when they grew up, I learned that when she became mother, I thought, oh yes, she's female. And when I saw the other one, which was black cat, it was male and it was running around, secretly here and there, doing the predation of yes, errors during the development, rare errors could be rare genetic disorder, epigenetic disorders. So during this part, during this trajectory, if the DNA is demethylated and it is demethylated, two kind of errors can happen. Demethylated DNA is prone to mutations, genetic mutations, and reestablishing and erasure are prone to epigenetic errors. Some certain regions which are supposed to be methylated didn't get methylated properly, or vice versa. Some certain regions which are supposed to be erased are not erased. The origin of this is could be genetic. More common the changes are, more common the diseases are, more rare the changes, more rare the diseases. Okay, so CalicoCat are females, and people knew about it. So now we know that this happens through X inactivation, because let me first tell you what is X inactivation. You have eggs, and you have one egg, and then you have a sperm. Don't go on site right now, I need to plot the chromosomes, and we know that X is inactivated in the sperm, and while it is inactivated because some chromosomes, some X, some sperms are Y sperms, some sperms are X sperms. If X chromosome is activated, then identity of sperms will be different. You will not have X is inactive in the sperm. So fusion happens and we get this zygote. So one hypothesis says that we inherit an inactive chromosome. Okay. So this is coming directly from here. But there is another hypothesis which says that this gets reactivation, and then reinactivation. At 4-cell stage. So nevertheless, both the hypotheses are out there, and this one is more popular. Then, so this is always paternal chromosome which is inactivated, and this remains like that in placenta. Oophoectodon and placenta is always the paternal chromosome which is inactivated. the you will have other development, right? And you in the end you are doing this ACM, trophoblast, right? So plesiocyte developing the trophoblast and trophoblast is also having inactive X chromosome from maternal. So, but here in this stage you have reactivation of both means what? There is only one which is inactive. Reactivation happens. Reactivation has happened during the blastocyst stage, and this will develop into placenta and the embryo, right? So here you have paternal chromosome which is inactive. At certain stage in the post-implantation embryo, I don't recall at what stage precisely, you have inactivation now, which is random X inactivation. Okay, now it doesn't matter is paternal or maternal. Okay, this is what we said, mitotically stable random inactivation. And if that happens, okay, one of the X chromosome will be inactive. Whether it's maternal or paternal will depend on at this stage. Okay, let's say the skin is developing here. Okay, these are the cells where we had paternal chromosome inactivated. Okay, and then there will be another cells which will be maternal chromosome which is inactivated. Okay, so you will have different population, and as they spread, they proliferate over time. Embryo grows. What happens is that they form a particular organ. One of the organ is skin, okay, tissue. Skin is a tissue, and the depending upon what gene is located on the maternal chromosome. What gene was located? Let's say there was a allele here for orange color on maternal chromosome and for black color on paternal chromosome. And depending upon which one is active in which cell population, you will get orange color or black color, right? So, and then they, once it is established, or in the maternal silencing established, they will divide now, they will proliferate with that configuration. Okay, for example, it becomes orange over here, black over here, likewise. So you get these patches, X inactivation, and you know why X inactivation happens, because of the double dosage. This is compensation on, for example, males you have XY chromosome, so X is having one dose only, right? Females is double dose, you need to silence one of them. But there are regions on inactive X chromosome which are not silenced. What are those regions? X inactivation is not complete. There are regions which are escaping X inactivation. What are those? Anybody? You know, in males you have X and Y pairing. How does that pairing happen? Homologous pairing happens, right? In males also you have one X is small, sorry, Y is small, X is bigger. Still pairing happens. There is a region on X chromosome called pseudo-homologous region. Those are the genes which are expressed. So because it is expressed in males also, right? So that is expressed. And X inactivation has evolved in mammals, amphibians only, and like genomic imprinting has evolved only in amphibians. And both of them has some association with placenta. I gave you the example of imprinting that one because you need maternal and offspring collaboration and so on. So I could have given evolutionary viewpoint of these, that you have monotreme like platypus, and then you have kangaroos which are marsupial, okay, and how it has evolved, how X inactivation has evolved. X inactivation in kangaroo or other marsupials is completely different than mice, okay, or human. X inactivation is at very rudimentary frames. So that was all from my side, and if we get time after your journal club, I will try to, or somebody will try to present the overview of this course, what have we learned, try to streamline the things that now it has been scattered in different instructors. Hopefully we will do a better job next time. So if you have any question, we can discuss that now.
So I had two questions from this slide. So one is regarding the postnatal development. So here it is mentioned that the like sperm and ovum, the percentage methylation DNA is same. So
Oh yeah, no, that's
Yeah.
a mistake. Good. So this sperm thing, so that should match this thing, right? It should be higher.
Yes, sir. And so secondly, so I was just wondering this point. So here we have two spots where the demethylation is happening. One in the like inner cell stage and one in the PDC stage. So I was thinking like rather than going up and again going down in the first stage, in the 3.5 stage itself, can't the cells like
Oh, yeah.
cells segregate?
Yeah. So if you go and do literature search, you will find that some people have drawn a trajectory where they show that my PGC is just coming like that. Okay, you will find some papers. But I think that's not the popular view. It seems like there would be some cells which have fate to become PGCs. Okay, just like here, you have some cells which have fate to become ES cell, inner cell mass. There are some cells which go to genital reach, right? So there are cells which move to genital reach, and that movement happens here. These cells are moving to genital reach, germinal reach, whatever you call it, germinal reach in the embryo. Okay, if the cells which move to germinal reach has an environment to demethylate. Okay. Now, what do I mean by environment? And I think nobody asked that why this trajectory is going up and down, how it is going up and down. What do you think? Why these trajectories are going up and down? So, there has to be a cause. What's the cause here? I just answer that in case of germinal reach. It's the environment. It's the hormones. It's the, for example, female germline environment, the male germline environment, the embryonic environment, right? The morphogens, the hormones and all that. So it's epigenetic changes, if it is happening, it has something to do, always remember, it has something to do with the environment. It has to be some signal which should tell the epigenome. Yeah, so your question is right, that some people actually think that this is the case, the dotted line is the case here. However, popular view is that this is not the thing. Until the certain cells move to germinal reach, demethylation happens. These two phenomena, movement to germinal reach and demethylation, are synchronous. Yes.
And so the third one is like, could you say any example of the random monoallelic expression?
Yeah, there are. So I said 10 to 20% of the genes are random monoallelic expression with so many genes. It's not one gene. Many of those genes are also sensitive. Autosomal genes. These are autosomal genes, and many of them are actually related to development. I don't have specific. EPHX1 is one gene, because I worked on this phenomena, so I recall this name. There is an impact gene. Some people say it's imprinted, some people say it's a randomly inactivated gene. So that's also an example of random monoallelic expression. But there are quite many, thousands of genes.
So sir, at every stage, this monoallelic expression is happening random, right? So I mean, I don't get the difference between mitosis.
Before proliferation, some decision has to be made. For example, for X inactivation, some decision has been made, right? At this stage, if this particular cell over here has made a decision that all the daughter cells will be having paternal genome inactivated, X chromosome. Then all the daughter cells of this cell, will have that maternal genome inactivated. Okay. So the same applies to random monolithically expressed gene. Okay. So that decision has to be made before proliferation. Let us say you are growing the cells in a petri dish. You have grown, and from this clone you took some small amount of cells and you plated them here. Okay, so this all the cells which will make colonies here are actually coming from one or two cells only, right? But if that decision was made here that your paternal gene, certain gene A, which was uh, paternal, is inactivated, all the cells here will have that paternal inactivation. Daughter cells. So sometime in development, when, for which gene, what is the right timing, nobody knows. Okay? But in the mature tissue, okay, in the differentiated cells, if I take my skin cells or some cells I grow, I find that there are genes uh, which are randomly inactivated. So to find out whether this is clonally stable or not, you need to do these experiments. Take primary cells and do this experiment. Okay, you have to do this experiment to understand whether the gene that you're talking about is clonally stable. This expression, which is monoallelic expression, is clonally stable. Clonally stable means that all clones have same allele difference expressed.
So if this is true, this decision is made before the proliferation, then that is similar to mitotically stable, right? So I mean, I didn't get the difference between random and mitotically stable.
Random one is that let's say there will be no mitotic stability, which means that in this experiment it is clonally stable here. Then you learn that for in case of random stochastic monolithic expression, you will find that well, same A gene is actually now inactivated maternally. Okay, there is no rule. Maybe it's paternal inactivation here. A is paternally inactivated here, and it is maternally inactivated here. Okay, there is no rule. It is completely stochastic. In one cell, you have one allele, maternal or paternal, anyone. Another one, you have another allele. Or maybe in another cell, you have both of them expressed. There is another fourth cell where both of them are repressed. So this is called random stochastic monoallelic expression. It's really stochastic. In real sense, it's random. It's not maintained. It's not maintained. So you can think of three hierarchies. One is completely random. The random in layman terms, it's really random. Second one is mitotically stable, or we call it clonally stable. And the third one is parentally fixed, genomic imprinting. Relation to genomics. Yeah, Yudish, you have a question?
Yes, sir. So this is like after like in an overall point of view. Sir, one example that I found striking where the Bayesian architecture plays a role like indirectly and not on gene expression when the heterochromatin is on the outside and periphery, and there is this hypothesis that it guards the inner active chromatin from insult, right? Like ionic and non-ionizing radiation. So it protects in that way, right?
Yeah, so as I mentioned, so there are two things here. First is that so this is your associated with the very body, right? And then you have chromatin inside. So you can have this, let's say, UV light, and which is present, and it turns out that it can, you will have mutations over here, you will have retrochromatin, okay? And recombinant is more important. It's actually secure, right? It's working like a sunscreen. Okay? You have certain screening around the eukaryote. I think eukaryote is the one which is the most gene dense one. Most of the genes are actually located in eukaryote, not in ectochromatin. Ectochromatin is enriched in non-coding regions. Okay, so which is like a you can think of a sinkhole for mutations. Okay, that's one reason, but that's not the only reason. You know that eukaryotin replicates early. The early replication in S phase, okay? And this replicates late. And what happens is that during the early replication state, early S phase, you have ample amount of replication factors. Okay? But as the S phase reaches towards end, there is a stochastic depletion of certain replication factors, okay? Or the DNA repair factors. You can think in that way. DNA repair factors will be depleted, or replication factors will be depleted, due to which you will have certain mutations accumulated in the ectochromatin region, late replication. So it could be because of replication also, okay? Not only the physical protection of eukaryotin and absorbing the mutation from the periphery. So both the things are possible. Some people have tried to negate this one. Personally, I don't entirely agree that replication has no role in that. It is known that late replicating regions are prone to more mutations, accumulate more mutations.
So I have two questions regarding this. So first one is, so we don't know whether this is causal or is it a consequence of replication, right? It came first and
No, we can't say that the mutation is causing late replication. Right? Other way around is not possible. Mutation doesn't cause late replication until or unless you have mutation in a very specific region of replication region. Right? So it is random, it is unlikely to happen at a region given the long length of DNA. So I would say that, of course, establishing causality in biology or in any field is a big challenge. Okay, so whenever we talk about cause and effect, one has to be very, very careful. Most of the time, we are only talking about associations, okay, non-random association. Even if you say that you have a knockout of a gene and this is giving you this phenotype, so this is causing that. No, where is the causality? What interaction of this gene with other genes is actually causing that? This gene is having 100 interactions inside the cell. Which of those interactions, which of those functions are compromised? So if you really want to find out precise cause and effect thing, it's very difficult. Here also, these are associations.
the second question is, if higher order arrangement of chromatin, does it intentionally or unintentionally influence the way nuclear synthesis process?
Yes, it does. It does. There's something called mechanosensing, mechanotransduction. If you have, let's say this is a cell population. In tissue or in a dish, you have certain cells which are located towards the interior of the colony, right? You have these cells which are located towards the interior of the colony. They have a different environment, while these cells have different environment, right? And not only the chemical environment, the physical environment is also different, because you have some extracellular matrix here, right? You have some other environmental stress here, something which is attached to some other tissue or something else is around here, and likewise you have direct interactions here inside the tissue, and also eventually the different environment. Not only the chemical environment, but the physical environment is different for each cell. So there is something called individuality of cell. Each cell has its own identity because the expression level at single cell is different for different cells. Okay, if you take a population of cells, you get the expression of genes will be different for different cells. Each cell has its own individuality, influenced by the environment, the one, physical or chemical. Second, stochastic changes during gene expression and replication. There could be stochastic error during chromatin remodeling, during expression and multiplication. Because of those factors together, certain genes are expressed or mutated and so on, right? Late replicated, because equals and so on. So yes, if you use a very tight matrix, let's say you're doing your cells in dish, okay, and you have a matrix here, some kind of extracellular matrix, laminins and collagen and all that, and you control the elasticity of this matrix, collagen filaments and all that, and that elasticity will tell what will be the outcome of cells. Let's say you grow the stem cell, proliferate stem cells in different elasticity of matrices. Okay, if it is less elastic, you can the cells may differentiate in certain cell type. To change elasticity, the cells may differentiate in different cell type. So we have phenotypic association with the physical environment, and that could happen in this way. One example is that, let's say these two are the stem genes. Okay, have lesser expression and more is your transcription factory, but there was some mechanotransduction that they are actually pushed into this transcription factories now. They weren't expressed, right? So it can be some phenotype. Yeah, the short answer is yes, the physical environment matters.
So it is not only on a tissue level, but on an individual cell level also, the force.
The force travels, you might wonder how it is going inside the nucleus. The force travels through, let's say there is physical stress to travel through the actin myosin filaments to the nuclear structure. Has been shown how that this happens.
So has there been consensus to live following portion of answer like assignment you were yesterday?
No, actually, today I was busy in the meetings. He's also supposed to evaluate your answer sheets, and he's not only access to the class, multiple issues. I will raise this issue after the class. I'll call Shravan first, because I think in my absence he was coordinating the course. I'll ask Shravan to do that, and if there is no time to conduct any such exam or assignment, then scale your marks.
Yes, sir.
Excuse me, sir. So, sorry, just to follow up. So, this relation that you said between protein, DNA, and RNA, does it work both inside out with an outside?
Can be. For example, let's say there is a certain expression due to chromatin conformation, okay, and that chromatin conformation is a result of stochastic. Let's say some chromatin remodeling was erroneous in one cell, very different from this cell. So in this cell, different kind of genes are expressed, and if those genes have potential to impact the matrix, extracellular matrix, let's say that gene was collagen, the extracellular matrix is impacted now, right? So if that is impacted, it impacts the cells back. Yes, it is possible. Because the changes inside the chromatin could also be intrinsic changes, not always coming from environment. Intrinsic variation. Biology is not perfect. Hence, our body and cells are always experiencing errors. Various kind of leakage of RNA polymerase, DNA polymerase, and that chromatin remodeling can happen properly because it needs energy. Is that all? Okay, so I will, for both the lectures, I will put up these slides together. One more question and I'm stopping here. Your journal club will happen physically. You know this, right?
In hybrid mode, right?
Yeah, hybrid mode. But the presenter has to be here, physically, I think so.
Yes, sir.
And then you have presentation per day. How is that happening?
Yes, sir.
Okay. Okay, then let's stop here.
Recording stopped.